Good morning students, welcome to LEELA'S tutorial. In today's class we are going to discuss about embryo collection and transfer. So how we are going to collect the embryo from the female animal after fertilization. So here you can see it is the superovulation of the cow to produce more than 3 oocytes. So superovulation already we have discussed in the last class in detail. So in case of superovulation it is also referred as superstimulation, where the animal is given the increased hormonal concentrations to increase the gonadotrophin so that it will produce more number of eggs per estrus cycle. Right? Normally only one egg is produced from the follicle, but here they are going to produce more than 3 eggs. So here by means of superovulation, why you are going to do this in animals? Because in order to get the superior quality animals. Right, you want the superior quality animal to produce only the superior quality progeny. So now if you are going to go for the normal crossbreeding or selective breeding, the progeny may not receive all 100% desired genes from the parent. Right, so here in order to avoid that, you want the complete genes to be inherited from the superior animals. So in that case you are going to select the superior animal and also give the superovulation. So instead of producing one progeny at a time, it can produce 3 or more progeny. Right? So let us see. First one is superovulation to produce more than 3 eggs or the oocytes. Once the oocytes are produced, so it is occurring inside the body. Right? It is not in vitro, it is occurring inside the animal body. You are giving superovulation injections to the cow and allowing it to produce more number of oocytes. Right? After that, it is the artificial insemination. Again, this also we have discussed in last class, where you are going to collect the semen from the bull. Again, this must be the superior quality bull, right, where it is having superior quality genes. Right? So again you are going to select the bull and collect the semen and then separate the viable or you can say active sperm cells. Right? The active sperm cells are separated, then you are going to inseminate them into the vagina or the cervix of the female. Right? You are introducing with the catheter. Right? The sperms are introduced with the catheter into the vagina or the fallopian tubes or into the cervix of the female reproductive system. Right? So here the fertilization is occurring inside the body. Okay? So it is called as artificial insemination. Right? You are injecting or introducing the sperm cells with the help of catheters. Okay? So then it is allowed to get fertilized or get the egg cells which are fertilized inside the cow. Right? They are allowed to develop for 7 days. Once they get developed, so again in embryo development we have seen it is a single cell stage, 2 cell stage, 4 cell stage and 8 cell stage. Right? So at the 8 cell stage or the next stage, the blastocyst stage, right, you are going to recover the embryos from this female animal. Right? So now the embryo recovery with the help of veterinarian, right, by using the catheter and the recovery fluid, they are going to inject the recovery fluid into the uterus and then with the help of catheter they collect the embryos. Okay, so now it has produced more than 3 oocytes. Right? If it is producing 4, then the embryos will be 4. Right? If it has produced 5 or 6, then again embryos will be 5 or 6. So all the embryos will be collected by using the catheter. Once they collect the embryos, now it is the introducing the embryo into the cervix of the or into the uterus of the animal. So here one animal cannot bear all the progeny at a time in its womb. So what they do, they extract all the embryos but they introduce 1 or 2 or at the maximum 3 into each animal. right? They may introduce these embryos back into the same cow, or they may introduce two embryos into each cow. Okay, so that they will bear the baby for gestation period. Okay, so it is embryos are introduced passing through cervix into the uterus using long and thin catheters. Right? So once you introduce into the womb of the female animal, they develop into baby by completing the complete gestation period. Right? So here like that, each cow at a time can produce only one egg, that means only one calf. But in this case you can see if it has produced 5 eggs. So the progeny is also 5 at a time. Right? Though the actual cow is not bearing the babies or the calves, they have introduced the developed embryos into the another animal or the surrogate animal. Right? The surrogate animal is only bearing the baby for the gestation. Okay? So here the superior quality cows or the progeny are obtained at a time. Is that clear? So at a time you can produce more number of superior quality animals. So this is a main advantages. This is one of the artificial breeding technique where you can produce the superior quality calf, whether it is a single or twins or triplets, whatever you want, how many embryos you introduce into the surrogate animal, so many calves or the progeny can be produced. Okay, in next class we will continue. Thank you.